technique?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, thank you. So let us start with the technique, the first technique to study the gene expression. So that is SAGE. Okay, so what is the full form of SAGE? It is serial analysis of gene expression. Okay, so this technique is based on serial sequencing of a tag. Okay, a tag which is made up of 10 to 14 base pairs. Okay, and these tags are going to be unique for every gene. So there are regions in your genes which are going to be unique. That means that sequence is not going to be present in any of the sequence. So those unique tags becomes a kind of like identifying tags for those particular genes. Okay, so what they try to find out in this particular method is what is the absolute abundance, matlab exactly how much number of your mRNAs, okay, exact concentration of your mRNA, exact number of your transcripts that is expressed in a particular type of your cell. So, and one of the very important advantage of SAGE is that it does not require any pre-existing clone. मतलब, in case of microarray, we require probes. So probes हमें कब मिलेंगे? Only when we know the genes, right? Then only we will be able to develop a particular probe for that particular gene. But in case of SAGE, we don't require such kind of probe. We can directly work with the gene. So it is possible that we can quantitate or find out what is the level of new genes also, or uh, whatever your known genes अगर नहीं है, unknown genes है, तो उसको भी हम quantitate कर सकते हैं. We can find out how many numbers or how much amount of those genes are being transcribed. So let us understand the principle behind this SAGE methodology. So as I was talking about short sequence tags, that is your 10 to 14 base pair tags, they contain sufficient information to identify uniquely a transcript. Okay, so because they are going to be unique for that particular mRNA. Uh, so provided that tag is obtained from a unique position within each transcript. So sequence tags can be linked together to form long concatamers. Okay, concatamers क्या होता है? When you have many sequences joined one after the other. Yes, so there's a rolling circle we have seen that your viruses they replicate. So continuously जो भी तुम्हारा, so if you have 100 base pairs का the circular gene, so what will happen? It will go on 100 करके so लंबा सा एक gene बनेगा, right? So that is called as your concatamer. So in this case also, this whatever your unique tags are there, they are being formed, or they are being uh, they are linked together to give you a long serial molecule which can be cloned, and then we can sequence it, and then we can find out how many times that sequence is present in that long concatemer. So every time, so if you say that you find out a sequence 12 times, that means there are 12 molecules of mRNA in that particular cell. If I'm getting for 50 times, that means 50 molecules of that mRNA is present in that cell. So quantitation of the number of times a particular tag is provides the gene expression level. So that is what I was talking about. That how many times that tag is present in that long sequence. So that will tell you that how many molecules of that transcript is present in that particular sample. So that is the principle behind it. So let us understand the steps. I will tell you first in brief. Okay, try to just go in brief, only the major steps, and then we will go on understanding the entire process in detail. So if you don't understand here in the brief, just don't worry. We are going to discuss it further. So first of all, what you are going to do is you have to isolate the mRNA from the sample. So whatever is your sample, a tumor cell or a dividing cell or anything, whatever cell is there. So you need to isolate your mRNA from that. Once you have isolated that, extract a small part of the sequence from the defined position of each mRNA molecule. So from those mRNA molecules, you have to extract matlab take out, excise out. How can you excise out apart from any mRNA or anything? Using your restriction enzymes. Okay, so restriction enzymes will help us in taking out those specific parts, which are your unique sequences for every particular DNA, mRNA or your gene. Now, whatever these small sequences or small pieces that you have got, you have to just link it one after the other using linkers. Okay, so we will go in depth of these things, don't worry, so that a concatemer is being formed. Now you have to clone these concatemer or these chains into vectors and then put up in the bacteria. Once it has been done, you sequence these chains using DNA sequencing methods and process the data with computer to count the small sequence tags. How many these tags are there, you have to count. Okay, so this is the steps in brief. Now let us go in detail about the flow. So here what you are going to first do is isolate the mRNA. Okay, once you have isolated the mRNA, what you will do is you will add the biotin labeled DT primer. Okay, DT primer का मतलब क्या है? Poly T. Okay, poly T primer क्या करेगा? Yes, you know that you have every mRNA has a poly A tail. Am I right? Yes. So what will happen? This is your poly A tail. So just one second. Let me. Okay, I'm not able to see the chat now. So if you have any doubts, kindly tell me to stop and repeat. Okay. So you see, this is your poly A tail. So when I am using a DT primer, what will happen? So it is made up of poly T, right? So that poly T will go and bind over here, bind to this poly A which is there, right? So when it binds to this poly A, so it is a primer. Now what you can do is, so this is mRNA, was your single-stranded mRNA. So using this poly T, now using your reverse transcription, reverse phase, what I can do is I can synthesize your another molecule, right? So that will give you a double-stranded cDNA. So that is how your double-stranded cDNA can be produced. Am I clear? So this is your double-stranded cDNA that is being produced. So once that is being found, now what is the use of this biotin? Biotin की हमें क्या जरूरत थी? Because this biotin will have a affinity towards streptavidin that everyone knows. Biotin and avidin have in affinity towards itself. So now you will be having your streptavidin coated beads. Yes, beads लोगे जिसके ऊपर तुम्हारे streptavidin रहेगा. So streptavidin can go and bind to this biotin. So what will happen is now this entire cDNA will get bound to this particular bead. Am I right? Okay. Now you will be using an anchoring enzyme. Yes, an anchoring enzyme will cleave this cDNA at a particular position. यहाँ पे cleave करेगा. So just a second. Okay. So that is how you are going to get different fragments. Okay, you will be getting different fragments. So now divide into two pools and then you would be adding your linkers. Yes, so you can see over here you can have linkers. This is your linker molecule. Yes, so linker molecule. अभी linker molecule को तुम कैसे add कर सकते हो? Using your different types of see, you see over here what is the sequence, and वो sequence के हिसाब से you can have your linkers that will be attaching specific molecules to one end, ligated. Then what you will do is cleave with the tagging enzyme. So tagging enzyme is going to be different. Okay, so tagging enzyme से तुम जब cleave करोगे यहाँ पे it will cleave off. Now again you will combine this pool of these whatever your molecules that you are getting one after the other, and then what you are going to get is a dye tag. यहाँ पे दिख रहा है, you are going to get is a dye tag. ठीक है, so क्या हो गया अभी यहाँ पे? I'll just repeat. Okay, you have cut it over here. You have cut it over here. See this. यहाँ पे हमने जब इसको cut किया, anchoring enzyme से we have cleaved it. So because of cleaving, we are getting this end. Okay, we are getting this particular end. So now so you can see I am getting these loose ends, loose fragments. So using a linker which will have a uska end dekho. You will have the end. Just a second, this pen color. So now see the end of this linker. It is going to be matching to this whatever your overlap region is there. CATG hai, so yahan pe it is your GTAC, which is going to be, complementary to each other. So they will get properly ligated to each other. Okay, is it clear? Then next, once they get ligated, now you are using a different enzyme. That is your tagging enzyme. You are using your tagging enzyme. पहले क्या था? Anchoring enzyme. Then you are using tagging enzyme. So tagging enzyme क्या करेगा? It will cleave at some other region. Yes. See, and you are using this particular the same thing. The mRNAs are n number of different types of mRNAs. So you have say 50 different types of mRNAs. For all these mRNAs, you are giving the same treatment, because here we are not specifically using any primer specific for that mRNA or any primer or probe for that particular mRNA. All the mRNAs are going to have this poly A tail and they can bind to this. So the initial process, whatever we have seen, that entire thing could be done for every type of mRNA. Are you able to understand? So ऐसा अगर molecule मुझे एक मिलता है which is bound to a linker. Okay, so if I have 50 different molecules of mRNA, I will get just an arbitrary number, please understand. So I am going to get 50 different such kind of molecules. Am I right? And every time this end is going to remain the same for them. Yes, they will have same different ends. So when you are having a pool of it, you combine them together, this end can ligate together. Yes, you can see. So ये एक molecule दूसरे enzyme से मिल रहा है. ये एक molecule you are getting from one mRNA. This molecule you are getting from another mRNA. Now they are combined together because they are being cut with the same enzyme. So same complementary ends would be produced. So this becomes a ditag. Now what you will do is, if you are again cleaving it with the anchoring enzyme. So remember what happened? Anchoring enzyme का कहाँ हमने role देखा था? यहाँ पे. That when it cuts over here, it gave you this particular sequence, right? This particular whatever your ends were there, and using the same ends, your linker was added, right? Using the same end, your linker was added. Now, if I again use the same anchoring enzyme, what is going to happen? यहाँ पे जो तुम्हारा link हुआ था, वहीं पे again cut will take place, right? So that is how you will be getting your such kind of your ditag. Okay, you will get such kind of ditag, and all the ditags would be amplified. So they would be amplified and then they would be connected to each other. I said all the ditags could be ligated with each other. Now, is it easy to ligate the ditags? Yes, very easy, because each of these ditags will have this as your end. So all are your cohesive ends. So they can be easily being connected to each other and then sequenced and recorded. Am I very much clear to all of you students? Understood? Have you understood this process? This is the overall method. This is the overall method how your SAGE takes place. Okay, so we will be going in depth of this method. So first, as I told you, first is your trapping of RNA with the beads. mRNA ending with long strings of adenine, poly A tail, as I've already told you. So it consists of 20 or so DT acts as an attractant to capture the mRNA. So you can have, see, there are two ways. Either जो हमने वैसा देखा कि you can use a poly P tails which is bound to your biotin, and then biotin, which or streptavidin, which is bound to your beads. So that is how they can attract onto your whatever your mRNA is there, that can bind to the beads. So you can see over here, or else directly the beads can be coated with poly T, and then the mRNAs can bind to it. So finally, what is our role? The first step, that you should have beads with the mRNA on its surface. Then you will, using your this DT primers, the cDNA synthesis will be taking place. Okay, so that's how your cDNA synthesis will take place with your biotin labeled primers. Yes, so see, this we have seen. Then what will happen? The cDNA is cleaved with the restriction enzyme. So an example of your restriction enzyme is your NLA3. Okay, so average length of cDNA is your 256 base pair with sticky ends, which are being created because of the cleaving of this. Yeah. So this is your anchoring enzyme. Please understand, this is your anchoring enzyme. See, this is your cDNA. वो cDNA को अब क्या करेंगे? You will have, let me go back to this format. So you will have this cDNA. When you are cleaving with this restriction enzyme, it is going to cut it like this. Yes. Then ligation of the linkers to bound to your cDNA. So captured cDNA are then ligated to linker, because we have already seen that you have this cohesive ends. So those cohesive ends will help us. So you see, these are your linkers. So using these linkers will also have the same ends as your NLA3 restriction enzyme sites are there. So those same cohesive ends would be there for these linkers also. So that is how you would be binding to it. So if you say this is your pool A and this is your pool B, depending upon how many mRNAs that are binding to a particular bead. So once that is being done, now you will be cleaving with the tagging enzyme. So usually it is your BSMF1. This is the enzyme that will cleave, releasing the linker adapted whatever your tags are there. So you can see over here, if I'm cleaving using this, if this is being cleaved, you understand the same thing I have discussed already. So यहाँ पे तुमने cleave किया, so you are going to get such small molecules with the linkers attached to it. Yes, with the linkers attached to it. Then formation of ditags. So what will happen is, I have already told you that these linkers would be having again your, with the help of your ligase, they would be connected to each other. So you can see over here, this is one type of your mRNA, this is the other type of your mRNA. So maybe both of these are connected together and you are going to get a ditag. So such kind of ditags are going to be obtained. So these linker ditags constructs are amplified. Many PCR molecules are being produced. Essay. So same molecules, many copies are being done. Then isolation of this ditags, the cDNA is again digested, again with the anchoring enzyme. So we have seen, अगर anchoring enzyme से digest करेंगे, तो क्या हो जाएगा? Here again there would be a cut. These would be cleaved. These portions are going to be cleaved. So this portion is going to be released free. The linkers are released free, and this ditags are going to be released free. So once the ditags are free, again they are going to have this sticky ends or cohesive ends, yes, present. So these cohesive ends which are there, that will help. So if that cohesive ends are going to be there in this also, this also, this also, this also. So in all the molecules they would be there. So a concatemer, that means linking of this ditags will take place. So once that is being done, lot of copies are required so that they can be inserted. So they are inserted into your bacteria. So million copies are being produced. And then what is going to be done is, how many number of tags which are there, which come from same molecules, that is being understood. So how that can be done? You can go for sequencing. So after sequencing, you will understand how much number of this tags is being done. So I'll just explain for 2-3 minutes. You can see over here, this is how the data will look like. So this is a tag, okay? So what is the count? How many times that tag was present in that concatemer? वो दिखाएगा. So इसमें से, तुम अगर देखोगे तो you will understand. See this first one, it is showing you 1283. So definitely the amount of mRNA for this particular tag is much more. So see this particular thing, last one. So first or last में देखो, last वाला is 110. So that means stored in your gene banks and all these things, okay, so which have the record of all these tags that can be stored. So using these phage we can analyze the difference between gene expression patterns of cancer cells. Sorry, again I got disconnected. So we can study what is the gene expression cells, gene expression of different cells. Yes, examine the transcript which is present in the cells, number of mRNAs which are there, compare between different types of cells. So that is what, as we have seen in case of different gene expression ka jo applications hai, the same applications are going to be there for your phage also. So it will give you a profile of the healthy cells and the disease cells. Yes, it will try to understand the downstream targets of oncogenes and your tumor suppressor genes. That means because of these oncogenes or your tumor suppression genes, which are the mRNA which are overexpressed or underexpressed, that can be understood. So one of the biggest advantage, as we have already seen, that we did not have a prior knowledge about these variants or these transcripts. Unknown ones can also be discovered, and direct counting is possible because of this particular method. The problem, limitation of phage, sometimes the length of the tag is extremely short, and so if the tag is derived from an unknown gene, it becomes a little difficult to analyze or identify which is that gene. And type 2 restriction enzyme does not yield always the same length of the fragment. So that is also a problem. And as we have already seen, always mRNA expression and protein expression may not be correlated. Okay, so that is all about phage. Please tell me quickly in the chat box, have you understood this or not?